is Tuesday, August 18th, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech. We are in the business of mapping the early stage signals inside of the incredible science space before they become big news. We focus primarily on therapeutics. We're so thankful that you've joined us today. Jeff, no guest today, but there's a boatload of signals to get through. You and I had a very chaotic and busy last week, so lots of cool science to get through, especially inside of the therapeutic space.
Yep, more neurology.
Exactly.
Neuro is blowing up lately. And a few new companies, at least one like new company out of stealth.
Yes.
And some pretty big Series A's. Yeah, got a lot on the dock. Hopefully my stupid computer behaves today. I'm already seeing some glitchiness. I've really got to figure out what the issue is with this thing. It's not doing well at all. Let's try this instead. See if that helps. Anyways, yeah, no, lots to talk about, and looking forward to discussing it with you, Benjamin.
Right on.
Also, do you notice anything different today?
I was going to point that out. Jeff, you're dressed up today. You must have been at a wedding.
I can't have you wearing a suit every day, and I'm wearing like a black t-shirt that smells like dog fur anymore. So it's time that I came to work wearing professional attire for once in my life.
Right on. Excellent. As Andy Lewin over at Lentitech would say, standards need to be kept up. Shout out to Andy Lewin.
Your posh Britishman accent is getting very good, Ben.
Thank you. I have spent far too much time in front of the mirror practicing impression.
Yeah.
Excellent. All right, so let's just quickly go through the leaderboard today here, Jeff, before we get into the signals. Obviously, there's going to be some overlap here. But yep. Leo Therapeutics right at the top with a boatload of signals. Let's go.
Yeah. So I think that they're the first one up, maybe. Oh no, Boulevard Bio's first. But yeah, Leo Therapeutics, neuro metabolomics based therapeutics. Really cool stuff. They had a Series A with Eli Lilly included, and initiated a phase 1b/2a trial in schizophrenia. So crazy, dude. I forget when it was, but like the first schizophrenia drug came out a few years ago, maybe, and I was like, you can make drugs for that. Like we know how it works enough to make drugs for it. Oh my gosh. So we'll discuss that and how those kinds of therapeutics work. InduPro. I think you picked InduPro, maybe.
I did.
But yeah, InduPro, let's see what they got going on here. Dose first patient, bispecific antibody drug conjugate. Must be an oncology play. And their Series B led by the Column Group. Nice. I'm not super sure what InduPro is doing, so I'm looking forward to getting your perspective on that. Aligos, we talked about them last week, I think. They maybe initiated some clinical trials potentially. Yep, dose first patient. And then Boulevard Bio, big coming out of stealth last week. Deerfield like incubated company or whatever you want to call it. So yeah, got lots going on. This platform is so cool. Oh, I gotta log in. Oh my goodness.
Well, while Jeff is logging himself in, let me tell you about TetriBio. It's all about who you know, but now you can catch prospects the moment they're ready, deep company research, live signals from your socials, verified contacts, and a whole lot more. Designed for biotech by biotech. That is TetriBio. Excellent. All right, Jeff, let's jump into the first signal of the day. Shall we get into Boulevard Bio here?
Mm-hmm. Okay, so Boulevard Bio.
Take us through it.
I got to figure out the zoom in depth here. Anyways, okay, so bispecific T-cell engagers. So I guess it's a tri-specific antibody. They're going after deep B-cell depletion. So Ben, and I'm going to guess it's for autoimmune disorders. It's got to be for autoimmune disorders. So yeah, so the idea, remember like when the first CAR T-cell thing came out, like CD19 targeting CAR T-cells for autoimmune disorders, and there was this whole thing about the immune reset and like B-cell depletion, Well, we can now use T-cell engagers to do the same thing, right? And instead of targeting just CD19, you also target BCMA. And we've seen a few of these come out recently. This CD19 and BCMA targeting T-cell engager, which is the CD3 part, is the T-cell engager portion. So Ben, do you know why you would want to target CD19 and BCMA. Or you know what? I'll ask you this question. When you see like a bispecific like T-cell engager for cancer, what is the purpose of the bispecific nature? What are they trying to overcome?
Just the heterogeneity
Right.
component. Yeah.
Mhm. So you want to kind of catch antigen escape, basically,
Sure.
right? But in autoimmune disorders, there isn't this issue with like the rapidly evolving population of cancerous cells. So what is the purpose of doing a bispecific strategy for autoimmune disorders, do you think?
Do you have more than one subpopulation that you're trying to go after while sparing other ones to keep them intact?
Basically, yeah. So CD19 is present mainly on like B cells, right? BCMA stands for B cell maturation antigen, and it's mainly present on plasma cells. Plasma cells are the mature B cells that actually express and secrete antibodies, which is what's causing the issues in autoimmune disorders. So by targeting both CD19 and BCMA, you can target both the immature quote unquote B cells and the mature plasma cells at the same time, leading to basically deeper B cell depletion and more durable remissions and better responses. So that's what they're doing. This is interesting to me. I remember when I read the press release, this MTS 128 here. MTS is not like the standard like abbreviation you'd expect for a company called Boulevard Bio, right? Like BLVD 201. So I'm pretty sure that they licensed. Oh yeah, duh, it's right below it. They licensed it from Biotech Bio, which I think is a Chinese company.
Is this a recurring? Is this a theme now? People are actually, instead of calling their companies like Metis Biotech, it's now Metis Tech Bio.
I've never seen one like this before. This sounds very Asian to me, to be totally honest. However that comes across, I'm not super sure, but it sounds very Asian to me. I'm gonna guess it's a Chinese company. This is like I've never seen a Tech Bio before. This is my first time.
Well, that is something I've heard inside of the milieu of biotech, right? People saying, "Hey, we need to start actually reframing certain components of the industry as tech bio versus biotech versus biopharma. We need to kind of have delineation of categories here. Tech bio is something, significantly different from biotech or from biopharma. So we'll see."
Okay.
Yeah.
If you say so. To me, it's all the same kind of stuff, I guess. But yeah, emerged from stealth, backed by Deerfield, which is a massive. Deerfield's like they're a VC, but they're more of like a company creator model, right? You see a lot of like, oh, it's a Deerfield company. So yeah, pretty cool. They came out of stealth, I think, like last week. What was this?
Oh wow.
Yeah, so like last Wednesday. So we didn't have a show on Thursday, so I figured it's a good time to talk about this.
Should we use the gong for any company that comes out of stealth?
We should have like a ninja built to change the course of B-cell driven autoimmune diseases. Yeah.
Okay,
cool.
so they are focused on bispecifics. This is a bispecifics play.
Yeah. Yep. They must have this is for what? Kidney disease, chronic neuropathy. Oh yeah, kidney disease. They've got a few in the pipeline, but their big one right here is this trispecific CD19 BCMA CD3 T-cell engager for autoimmune disorders.
Okay.
Mm-hmm. And they along with this emerging from stealth, they initiated a phase 1 trial in healthy volunteers.
Wild. How much money have they raised?
Oh, probably a boatload. So they got 65 from Deerfield, but 130 total across, different investors. Yeah.
Phenomenal. All right. So a well-positioned company. They've just emerged from stealth. They've got $130 million. They've already initiated phase 1. Yeah. Makes sense that they are at the top of the Biotech Voyager.
Oh, yes. And the big thing here, is that the CEO, led by Mahesh Karendi, Omega Therapeutics CEO, took the company public. I think that he was also involved in some significant like exits, but maybe that's somebody later. The $1.6 billion China in-license event for the trispecific and the Deerfield incubation model generally worth okay. Yeah, they in-licensed that tri-specific from this Chinese Metis Bio for $1.6 billion, like in total in milestones. So that
Whoa.
now in IND enabling, but they're already in clinical trials. They began dosing healthy individuals. So
Well played.
yeah, keep your eye out for this one. It's always really nice to see new companies coming out. Like it's always, I don't know about you, but I'm always just like when I see it on like when I see it in the press releases, like, oh, emerge from stealth, I just get a little bit of like an endorphin kick. Like, oh, biotech is growing.
Love it. Excellent. Okay. So, all that being said, Jeff, shall we move on to the next story of the day, which I believe is Artello Biosciences?
Oh, they've been busy. What do they do? Oh my gosh, look at this. What are they doing?
I saw that this morning. I was just like, "Oh my goodness, what in the world?"
Yeah.
leave some attention for the rest of us, please. But yeah, an absolute flurry of press releases from Artello. A lot of it kind of linked around a key patent that they've got running through to 2041. So that gives them lots of runway, right? That's 15 years from now. So they've locked in a key technology that should last well into our late 40s, Jeff.
Dang, we're old, dude.
We're so old.
Oh, man.
We're not even that old, but we're just that close. But anyway, yeah, wanted to spend some time on them today in particular. Oh shoot, I guess I'll have to add that image in again because I wanted to jump a little bit.
Oh, this is the cool linker technology.
Yeah. So if we just back up for a little bit here, right, and we look at what they're focusing on here. So what's really interesting with this company, they are focused a little bit more on an interesting application that I wanted to highlight today. So again, they are a clinical stage pharmaceutical company, but they are developing proprietary therapeutics, which kind of makes sense given the patent that they've been able to lock down, to modulate lipid signaling pathways. And the way that they're doing this is that they have basically developed a specific inhibitor that's able to get conjugated to lipids and kind of hack some of that machinery. So if we open up that picture that I've got here, ideally we can present this. So this is kind of what we're looking at. So normally you have a drug that's going to be relevant to whatever specific disease that you're going after. And basically they've developed a way to link the specific drug to a bile acid. And by being able to link it to a bile acid, you've basically got a bunch of these different pathways and like transportation mechanisms to allow you to get across the cell membrane into your specific cells that you're interested in going after. So that was just kind of cool, right? When you're thinking of some of the different ways that you try to get a therapeutic inside of cells and then targeted to a very specific area, they basically have developed what seems to be a fairly novel new way of basically unlocking a new transportation mechanism into the cell to go to a very specific destination.
Interesting. Yeah. Like transmembrane diffusion of drugs is actually a relatively rigorous area of study, but it looks like they're leveraging like bile acid transporters on the surface of the cell, and
exactly.
Like if a certain kind of cell type has more of them, you can have a higher concentration of the drug within those cells. So I'm wondering what kind of cells would have bile acid, like high bile acid transporters in the brain. It's for like pain, and they have one for anorexia and stuff. Like that's wild. Yeah, like a peripheral acting non-opioid analgesic targeting chemotherapy-induced peripheral neuropathy. Cannabinoid receptor agonist for cancer, anorexia. Oh, oh my gosh. We covered this company a while ago. I had to look up what this cachexia is. It's like when you get cancer and lose your appetite and you like lose a bunch of weight. Like chemotherapy induced like anorexia, basically.
Right. Well, no, the interesting component here, right, is not only do they have a somewhat novel delivery mechanism, but they're also combining this with like cannabinoids as well, which is just kind of like a couple different interesting technologies coming together that, I love very much, Jeff, which is stacking different
Pot.
stacking different technologies on top of each other.
Yeah.
That is not what I said.
Confirmed.
For the record.
Ben loves cannabinoids, especially the ones that you smoke.
That is not true. My wife doesn't watch this show, but her dad is an ex-cop, so
Yeah, she's doing this. She's like, I've had enough of this marriage, Benjamin.
Should we clip that? And let's not.
We're no longer married, Ben. Clip that, Isaiah.
But no, it's interesting, right? Because I believe, if I remember correctly, this company is also going after basically another challenge, which is like, the whole non-opioid space, which is, again, something that's really interesting inside of, like, you have cannabis, sure, and everyone knows cannabis because you smoke it, but there are all sorts of really interesting rare molecules inside of cannabis called cannabinoids that have all sorts of really interesting applications inside of appetite, what am I trying to say?
Suppressants?
Yeah,
Or like
suppressants.
agonists?
Yeah, agonists, like appetite activation, but then also pain relief, right? And so being able to find one of these very unique little cannabinoids that's at a very small quantity inside of cannabis, and then identify that molecule, pull it out, manufacture it, stick it conjugated to a lipid, allows you to potentially develop a really powerful application to go up against some of these opioid painkillers, and kind of provides an alternate way to deal with pain for folks.
Yeah, it's kind of like giving cancer patients the munchies.
Yeah.
That's basically what it's doing. You're taking the thing that in weed that makes you have the munchies, and you're giving it to people who are anorexic.
But as you'll note, right, when you go to that company breakdown section, it has applications in oncology, but also in pain. So the potential there being, not only will you have applications for giving anorexic cancer patients the munchies, but also helping them being able to deal with pain, and that's what ART 26.12 is, right? If you go down to their therapeutic assets, you've got one that's focused a little bit more on pain, and one that's a bit more focused on the kind of like the cancer-induced anorexia. So two really interesting applications, and they definitely merit their spot in the leaderboard of the Biotech Voyager for sure. Really, really interesting application. I'd never heard of this approach of linking together fatty acids with small molecules, but maybe that's just my ignorance, Jeff. The other thing that's interesting as well, when you go and look at that story, is like there's a special name for it, I believe. I was trying to find that, right? When you think of, or maybe I'm getting two stories mixed up. Maybe there's another company as well that we're talking about today that also deals with cannabinoids, because there's like a very special name for like this linking technology. Wait, I thought I had that. Yeah, they're called FABACs, or is that with a different company?
I have no idea what you're talking about.
No. So I'm getting myself mixed up here, Jeff. I'm getting this is egg on my face. I'm getting Artello mixed up a little bit with the next company that I'll be dealing with, which is Galmed. They're the ones who have this fatty acid combination. I feel like an idiot. My bad.
Well, hang on. This thing, so targeting lipid signaling pathways for pain. Oh, so they're just targeting the pathways.
They're just targeting the pathways.
Oh.
I got messed up. So
You've
I will come back
wasted
a little
all of
bit later.
my time.
When we come to the next story, I'll pull up the fatty acid conjugates again, and that's where it'll be interesting.
Interesting. Lipid signaling pathways. Dude, our cells are so insanely complex. Lipids aren't just like the membranes of things, which like it took me until like legit like my second year of grad school. It's so embarrassing to realize that like even organelles have membranes. It isn't just the cell membrane, right. But they're also involved in all kinds of like signaling pathways, which is like obviously, it's insane, dude. It's crazy how incredibly complex it is, but then two, how we can like discover things and then develop drugs to do stuff. Like what?
Yeah.
Okay.
And
Anyways,
then even the drugs that we develop, right, are such like a ham-fisted approach given the
well,
vast complexity
relatively
of the
speaking,
machinery. Yeah.
yeah, just like the hammer, we crushed it into enzyme.
Yeah.
Mm-hmm.
Nifty. Okay. That's all I've got for Artello, so apologies. I blame sleep deprivation
Well,
from
now what do
having
you
a
have for
2-week-old
GaleMed? The
baby.
same thing. It's just the same conversation again for GaleMed.
No, we're doing Leo Therapeutics next.
I know, but we're gonna get to GaleMed next, so you better get something going. Yeah, so Leo Therapeutics, $30 million Series A with Eli Lilly, and they've initiated a phase 1 clinical trial in schizophrenia. So I guess lots of neuro stuff. The thing is, dude, is like when you start a business, right? Especially like these days, now that we've seen a lot of the long timelines, the small patient populations in things like cancer and things like that. There are so many people who deal with things like pain, right? Or like other kinds of like mental disorders. Not that pain's a disorder, but it has to do with like your mental state. um, is my camera really, dude? We got to figure this out. What is going on with StreamYard? Come on, man. My computer cost $4,000 and it's like, oh, it can't handle StreamYard. Okay, anyways. Leo Therapeutics, metabolic therapeutics for high need CNS disorders. The idea being that brain metabolic imbalances drive the neuropsychiatric and neurodegenerative pathologies, which I think honestly, like this whole idea of like a chemical imbalance, not to be like controversial, even though I love being controversial. I've recently heard that a lot of this, like your chemical imbalance, was a bunch of bull. And that we don't really know what's causing these things, and it definitely isn't a chemical imbalance in your brain. So at least when it comes to things like depression and ADHD. But so brain penetrant oral small molecule that reduces excessive glutamate by inhibiting the mitochondrial enzyme glutaminase, targeting schizophrenia. Dude, okay, again, like the idea, like you just mentioned that that a lot of our therapeutics are like just like hammer and fist kind of thing. I feel again like I see so many glutamate targeting therapeutics that are just like targeting all kinds of stuff. Like this is a glutamate targeting therapy for schizophrenia. I know at least 3 companies doing glutamate targeting therapies for MS, right? So, and then they have LTX-002, which is an ASO that's targeting lipid metabolism for ALS. Yeah. So the cool thing here is that the CEO previously founded Prevail Therapeutics, which I think was exited to Eli Lilly, and now Eli Lilly is getting involved in their $30 million Series A. So watch for an Eli Lilly acquisition of Leo Therapeutics soon. It's my prediction. But yeah, $30 million Series A extension and phase 1 initiation. I'm actually very curious to see.
And you're right, Prevail was bought by Eli Lilly.
Was it? Yeah. So this is their phase 1 is in schizophrenia. Yeah. What do you know about schizophrenia?
Very little.
Me too. I know that I mean, like, obviously I know what it is kind of, but like, I don't think that I do. I actually saw a YouTube video one time of a guy who had paranoid schizophrenia, and he had some subversion of paranoid schizophrenia. And I expected to have like this person on camera just like completely freaking out and like being crazy. And he seemed kind of normal. And I was like, I don't know what schizophrenia is. Like, I don't know what it is. I always think of like just being a psych patient who's like hearing voices in the walls talk to them.
Right.
But that's obviously not what it is.
Yeah, there are some functioning versions of it, but yeah, you need some fairly heavy medication, and I think eventually it does kind of end with you going to some sort of psych ward. Like it generates over time.
It can get pretty bad.
Interestingly, the one little piece of interesting information I do know about schizophrenia is no one who is blind has ever gotten schizophrenia.
So you must need to see to hear the walls speak to you.
Something like that. Not to make light of it. But yeah, it is interesting to see the different applications coming out for schizophrenia.
Yeah.
More recently. I wonder what the catalyst has been in terms of the breakthrough there, because we went a whole.
Oh, you know what it is. You know what it is. There's two things. One of them is 2 letters, and the other one is time. The more time that goes by, the more we have time to research these things. And like, we just didn't know how the brain worked 10 years ago. And as you begin to understand more about it, and with the advent of AI, you can, we're going to see more and more of this. The brain's extremely complex, although most things are, but the brain is more complex than other things in biology. So I'm not surprised to have been seeing a massive wave of like neurotherapeutics, because we've historically not really had a whole lot of knowledge of how the brain works. So as we're getting more and more now, like, you're going to see a flood of therapeutics in the area.
Makes sense. Cool. All right. Shall we move on from Leo Therapeutics? A shout out to them. Are they at the very top? Are they at the top of the Biotech Voyager?
Yeah, they're number one. Number one.
Shout out to them. Let's give them a round of applause, shall we? Yes. Thank you for making it to the topic, the Biotech Voyager. You're super cool. All right. Moving on to the story I thought I was talking about. Which is Nurexone.
Oh, not Galmed?
Is it Galmed? Oh, we've got them backwards. Okay, here we go. Galmed. Here we go. And then Nurexone's next.
Dude, my computer just cannot handle doing this.
Okay. There we go. Perfect. Okay, so Galmed. This is, again, the company I thought I was talking about a little bit earlier. Apologies one and all, Jeff being the primary victim of my ineptitude. Really interesting company that just really jumped out today. So basically, this is a clinical stage biopharmaceutical company built around first-in-class synthetic fatty acid bile acid conjugates. So you've heard of, drug. You've heard of antibody drug conjugates. You've maybe even heard of degrader antibody conjugates. This is a fatty acid bile acid conjugate. So this is an FABAC. And so really interesting approach that I hadn't really seen before. I'd never heard of FABACs before. Maybe everyone else has. Really interesting application, and kind of the way that, and kind of how this works is, again, you have the ability to basically hack the transportation systems that are getting used for fatty acids to deliver your drugs. And obviously there's a couple different ways that can be really interesting, but in particular, just what is cool is the fact that this allows you to access some pretty discrete pathways that might allow for something that's a bit more targeted in your approach once you get inside of the cells, right? We kind of think of like small molecules or whatnot that just kind of like diffuse through, but there's not a whole lot of pointed guidance once you get inside. I believe this might be a far more what's the correct word? A far more elegant mechanism that's a lot more pointed to get a therapeutic directly where you want to go inside of a specific cell type. So in particular here for this one, they're going after hepatic fat accumulation and fibrosis in NASH/MASH, with the platform also being explored for gastrointestinal oncology. So pretty cool. And then they're also developing a 5 amino acid synthetic peptide targeting pro-inflammatory pathways in inflammatory disease. So this one's really cool. I had never heard of this approach. It's a company that's come out of Israel in Ramat Gan. And then just very recently, they just disclosed their preclinical results for this application inside of prostate cancer. So maybe with prostate cancer, there's some sort of preferential uptake of these types of like bile acids or whatnot. But yeah, it's still fairly early stage, but it was interesting just to see like a new, a new almost like asset class,
it says first in class. I mean, the thing is like, so fatty acid bile acid conjugate. Um, it's obviously a play on antibody drug conjugate, right?
Right.
The it's a targeting mechanism, right? It's the cells that have high whatever this receptor is going to have higher concentration of the drug. The same way that the cells that have a high concentration of the antigen expression for ADCs have a high concentration of the drug when you
Right.
give the ADC. Yeah, I mean, if
here's a question for you, right? They have showcased a 3 to 4x increase in kill of prostate cancer cells. Would this potentially be a route by targeting kind of the lipid side of this versus a particular antigen that you might go after with something like an ADC? Would this potentially be an approach that is more robust against immune escape?
No.
Why?
Because the way that the cells escape from like antigen escape is just like evolutionary. Cells that have lower expression of it, just like naturally, will just not get as much drug. This is the same thing. The cells that naturally express just a lower level of this receptor or this transporter will get less of the drug. What's interesting here is that it kind of bypasses the internalization kinetics of your target. So when you have an antibody drug conjugate, right? The antibody is going to have the drug conjugated to it, antibody drug conjugate. The antibody binds to the target. Okay, Ben? When the antibody binds to the target, how does that kill the cell? Right? With an antibody drug conjugate, the target is bound by the antibody drug conjugate, right? And then that whole complex has to be internalized.
Right.
Right. If your target doesn't internalize rapidly, right, the antibody could maybe fall off. You won't get a whole lot of accumulation of the drug in the cell unless the thing internalizes. With this method, you aren't relying on the internalization of the transporter. You're relying on the actual biochemistry that the transporter has been evolved to do, right? So the bile acid drug conjugate binds to this bile acid transporter, and the thing just pushes it through, right? And on top of that, when an antibody drug conjugate binds to a target, the whole complex gets internalized. That target's gone now, right? So as you have more internalization of those targets, the cell itself will become less and less and less concentrated of the target itself. But with this, the bile acid transporter just pushes the drugs in and it stays there. So you can definitely get higher concentration of the drug. And if they mentioned 3 to 4x, that actually seems kind of low. What are they comparing it to? Just like the naked drug? Or would it be like an antibody drug conjugate targeting this transporter? That'd be a cool experiment to do.
Yeah, good question. I'm not sure what I imagine they're just looking at like a control of just the drug by itself versus the fact that you've got a 3 to 4x increase in killing.
Yeah. It's in killing.
Kind of like
I wonder.
established.
Yeah, I wonder because the next question is, does the linker fall off eventually? And if not, does the presence of the linker affect the binding of the drug to the target? Right. I'm going to guess that the presence of that linker actually does reduce the efficacy or the potency of the drug itself. But if you can get more of the drug in the cell, then you can effectively inhibit more of the enzyme. So it doesn't really matter if it's less potent.
It could be. I don't know. They're fairly sparse on details because it's a new approach and they're just at the preclinical stage. But regardless, seems pretty interesting. And again, to some extent though, Jeff, with regards to like going back to kind of like this whole idea of immune escape though, I feel like a bile acid is going to be a whole lot harder to escape from versus a particular antigen that an antibody is going to be looking for.
I don't think so. How do you think like a tumor effectively undergoes antigen escape?
Well, that
Like
yeah,
the cells don't have a brain. They're not thinking, oh, they're targeting CD19. Better downregulate it. That's not how it works.
I get that for sure, but part of the reason, for example, like a specific antigen might be victim to immune escape, right? Is yes, to your point, right? You kind of have like just that like preferential, the preferential reality, right? That like the evolutionary component of this is something that's going to get cells that don't have this particular antigen are therefore going to be immune to the therapy treatment that's coming in. Therefore, we're going to preferentially increase that population. But like that's a specific antigen. With something like these lipid mechanisms, might this be something that's just more conserved and more required for the life of the cells versus the presence or absence of one particular antigen?
No.
Okay.
No, and if that was the case, then you can just target antigens that were like required for cell viability, because then you couldn't lose it, and if you do, you just die, right? Like there are those antigens. I don't think that this is like an antigen escape play per se. It's probably more of a drug concentration play, which is why they probably report their drug concentration. They're getting more drug concentration in the cell. It's a more effective mechanism than just like relying on target internalization. And it's likely that like bile acid transporters may be present at a higher level on the cell surface than most other antigens. So like you're getting more surface expression, no internalization kinetics dependency. One target can internalize multiple drugs. Like, so that's the big thing that I can see.
Okay. All right. Sounds good. But yeah, regardless, the fact is, even though they're at the early stage, taking an existing drug, right, Aramcol, and conjugating it.
No clue how to pronounce it.
Aramcol, I think it would be. To take an existing cancer drug and make it 3 to 4 times more effective at killing cancer cells seems like a pretty big signal. So excited
Yeah, really
to see where they
cool.
go with this.
Really cool. Where are they based at?
I believe they're in Israel.
Cool.
Yeah, there's a couple different companies that popped up today based in Israel. So
I'd love to get like Alan or like Liat on the show to ask them that question. Like, what was the therapeutic rationale? Is it I'm going to guess that it's a internalization kinetics thing and a concentration thing. So
shall we make a mental note to reach out to them? Be like, hey,
we shall.
We're fans. Tell us more about FABACs.
I can do this. I can add straight to Tetri.
Boom.
Bam. We're queued.
Yeah. Tetri.bio. Something that you can get yourself. A fully functionalized biotech designed SaaS for all things BD.
Amen.
Amen. All right. Very good. Perfect. All right. So Nurexone, what's up next here, Jeff? I believe it's InduPro.
Is that me?
I believe that's you.
Oh, no. Oh, no. I know what this company is doing. Okay. InduPro. What is the signal? Let's start here. Okay. $77 million Series B led by the Column Group, massive company, I think in New York City, the Column Group, PCG. And patient in their bispecific ADC IDP-001. Let's look at the indication real quick, because this will give me an idea of what this thing actually does. I think they're making proximity-based ADCs, which I think is like, it's an antibody drug. It's a bispecific antibody drug conjugate that requires both of the targets to be present in close proximity to like be internalized and like actually work. That's what I think is happening here. And we can discuss like the scientific rationale and like the possible benefits there. But first of all, massive Series B, well, not that massive, but about $80 million Series B. And the first patient, it's an oncology thing. And again, so it's non-small cell lung cancer. Did they disclose the targets? Because that will also tell me if, so yeah, EGFR, TAPA E1, what in God's name is this? Novel tumor antigen proximity antigen. I've never heard of this. Okay, so we're in for it here. So EGFR obviously makes sense. EGFR is extremely highly present on the surface of lung cells, lung cancer cells. So a lot of the EGFR targeting therapeutics are in lung cancer. But I've never heard of this, and I've never heard of doing something like this, which is why initially this is why this caught my attention. So let's just pop over to their website and see how they present this. Get closer to our groundbreaking work in proximity-based therapeutics. Let's see. Let's go to their science page. Okay, so we map cell surface protein proximity. This is one of those those areas of cell biology that like I never even thought of, right? Like you never consider like, oh, the proximity of these receptors on the cell surface. It's like, are they present or not? Was like basically the end of the line for the questioning. So the cell surface proteome exists as a multidimensional array of protein communities. I did not have that on my bingo card. Referring to the cell membrane array as the protein communities. By cataloging inherent proximity, we define protein spatial relationships to create novel therapeutic paradigms. Our high-resolution protein microenvironment mapping technology, that's a freaking mouthful, high-resolution protein microenvironment mapping technology feeds into our MINT platform and serves as a differentiating advancement over conventional approaches. Okay, so this is one of those things where like, hey, you don't want to have off-target effects, right? You can pick a target that's just not expressed on healthy tissue and is highly expressed on target tissue, but good luck, right? It seems like what they're doing here is they're saying, hey, we can actually have one more level of complexity here and say the targets can both be expressed on the normal cells, but they're highly expressed in close proximity on the target cells. Right. And that is the close proximity is actually like the differentiating factor between healthy and not healthy tissue. But I'm wondering like what is this
how does that help you though? Because even though they're in proximity, like with something like a bispecific antibody or something, right, where you're looking for those two different targets, how is it going to be able to differentiate between something that's super close together and something that's wide apart? Is that
I'm going to guess that both arms have to be bound to be internalized.
Okay.
Like it probably has to do with the biochemistry of the actual molecule itself. So
it's kind of an AND gate.
Yeah, it's an AND gate for ADCs.
Where's this company based?
And it's not even just an AND gate; it's an and proximity gate.
Right.
They are based in, I'm gonna guess Boston. Yeah, Cambridge.
Okay. I was gonna say, well, there's got to be some Spanish influence because they're calling tumor-associated proximity antigens tapas.
Is that Spanish?
Yeah,
I don't know.
That's like Spanish food.
Oh, okay. I mean, also it's tumor-associated proximity antigen, which is just like the natural. So I'm looking to see. They need a better website. What is going on here? Okay, so co-targeting to enhance ADC activity. So tumor antigen, tumor-associated proximity antigen. What are these? It seems to be like some kind of novel thing that doesn't even have like a real function, or at least the function's not important. Co-targeting to enhance T-cell engagement. Okay, so they're doing T-cell engagers too. Immune receptor agonism. Okay. So it looks like their moat is this MINT platform, this microenvironment proximity like thing, this thing here. So yeah, proprietary MINT knowledge base uses AI/ML to enable co-target selection. Okay, I'm gonna read through this because this is actually like incredibly interesting to me. Our MINT platform leverages deep learning analysis of proprietary protein microenvironment and membrane proteomic data to define the architecture of the cell surface proteome across different target classes and indications. This allows us to identify novel targets and co-target pairs for selective tumor. Okay, so I'm going to guess that it has to do with like single cell imaging at very high resolution, because you can if you stain for a bunch of different targets, right? We're going to stain for 50 like multiplex 50 different targets, right? And then do single cell like imaging, and then get like high resolution images of that, and then have AI/ML analyze those images and say, hey, look, these cells have target 14 and target 73 expressed in close proximity more than other cells do. Right. And then once you've identified that pair of targets, and it wouldn't even matter what they are.
Right. They can be anything.
Yeah. Yeah. That seems to be what it is. I'm wondering. I'm wondering if that's actually how they do it though. It's got to be like imaging, because how else would you do it? Oh my gosh, it's got to be single cell imaging. And this is the thing too. Like you see these. I didn't appreciate this concept until like I got into biotech, right? But you see a lot of these interesting like new research techniques, right?
Right.
And I'm just like, okay, like that's cool, but how could you ever use that? But I'm dumb. This is the problem. I'm dumb. And I wouldn't think to develop a proximity-based therapeutic class utilizing single cell high-res multiplex imaging. Right? Like I see like single cell high-res single cell multiplexing, and I'm like, okay, nerds. Like, what are you gonna do with that, nerd? It's like, well, we're going to develop proximity-based antibody drug conjugates and T-cell engagers. And it's like, oh, okay. You're not a nerd, you're a genius.
Yeah.
So here it is. Proximity-based drug delivery and proximity-based T-cell engager activity, which is super cool. Yeah.
Nifty.
Really nice. Also, my research came back from GalMad, 20 out of 100. Not good. Oh well, poor fit. Whatever. Okay, Ben.
Regardless.
What else we got?
All right.
Just a heads up. I might have to hop at noon.
Yeah. Okay.
Yes.
That's fine. We can run a little bit shorter today. So the next one up here is Nurexone.
Oh yes.
And then we're gonna have Feigns last.
Nurexone biologic.
Yeah.
Okay. What do we got?
So this is another company from Israel. And remember, Jeff, back a couple months ago, we talked about a company called Matriself or Matriself.
Yes.
They had won a pitch competition at ATW in San Diego back in January, February.
Yep.
So really cool at the time because basically they were going after spinal cord injury, and they were basically developing a matrix that's basically like a spinal cord implant that contains your stem cells that are basically going to help like reattach and regenerate your spinal cord. And we had this really cool little video that we showed on the screen of the rat that they were looking at that was, first of all, just kind of dragging itself along on its front paws and nothing else. And then after this implantation had taken place, they were able to use all four legs again. Super cool. And so they're driving that forward to the clinic. This company, also based in Israel, is focused on a slightly different approach. They're not developing like a matrix. They are actually developing an exosome-based therapy to go after central nervous system injuries and regeneration. So super cool. Like there's so much kind of coming out of that part of the world in the regeneration space for spinal cord injury.
So tell me more. How does this work? I get the matrix because the matrix, you can just kind of implant autologous like neuro progenitor cells basically, and just kind of like hope for the best. That's what seemed like Matrix was doing, but it seemed to be working. And you can kind of like cure spinal cord injury by having that matrix of neurons just kind of like create those synapses again. Yeah. How does is an exosome thing? So is it cannot be exosome based like neurotransmitter release. That would be too slow.
So basically what they're doing is they're using mesenchymal stem cell derived exosomes as non-genetically modified nanocarriers to deliver therapeutic molecules across the blood-brain barrier to sites of CNS injury.
Okay.
And so the lead asset of what they're actually putting inside of these exosomes is a PTEN targeting siRNA to promote neuronal regeneration in acute spinal cord injury and glaucoma.
It's an
siRNA that promotes regeneration in applications for spinal cord injury and glaucoma.
It's a P10 targeting siRNA intranasally to promote neuro regeneration. What is P10 doing that's causing the neurodegeneration to progress? P10 like promotes cell cycle progression, I thought.
Let's find out.
I used to know what P10 does. It's like a very common protein in cancer, but I've been out of the game too long. tumor suppressor.
So it
Oh, it's
acts
a
as a
tumor. Yeah, yeah,
major
yeah.
molecular brake on nerve cell growth. By blocking the mTOR pathway, P10 stops adult nerve fibers from regrowing after damage. Depleting or inhibiting P10 removes this block, reactivates growth signals, and helps damaged spinal axons.
Apoptosis. Oh, it's a cell cycle arrest protein. Okay, that I had it.
Yeah.
I had it backwards. Yeah, I'm actually very embarrassed. I definitely know what P10 does. So it must be
Mm-hmm.
involved in like suppressing the cell cycle in neurons pretty
Exactly. Well, right.
robustly.
mTOR, right, is like the master guider of all things cell health and
Well, mTOR is like metabolism. mTOR, if you have like rapamycin, like target of rapamycin, mTOR like just like promotes protein synthesis and like it's like a massive like grow. So if P10 is inhibiting like PI3 kinase mTOR activity, then yeah, for sure. So again, okay. Okay, let's be honest. A siRNA, bro, they've been around for like decades.
right.
This is not new technology. Why are we now seeing a bunch of siRNA based therapeutics like making waves? Like it's been a thing for a long time. I was going to go into my PhD in like 2016 to study like siRNAs and stuff, or like, well, small non-coding siRNAs. But
I feel
like,
like I feel like it's just a lot earlier than you think it is, Jeff, right? Like
we've been using siRNAs in like basic research for decades,
sure,
right?
it's way easier to knock in or knock out something in basic research. It's a lot harder to do that in the vast complexity of the
Okay,
brain.
but this
With
is where I think that the actual innovation has been taking place. Why is it harder to do it in the brain?
well, you've got to be very precise in your delivery mechanism.
Delivery.
Yeah.
It's a delivery innovation.
Yeah.
Right. And again, it gets to like the I'm dumb thing. I really wish it's funny how life works. Like I've learned so much in the past like 2 or 3 years. I wish that I could go back and know what I know now, but at like 22. Right? Like I say get a rewind, please.
Everybody. That's everybody, Jeff.
I know it is. That's why I say like life is funny like that. But I'm sitting here thinking like, dude, if I knew now, or if I knew what I know now then, gosh, I would bet on so many football games.
Welcome to the premise of Back to the Future.
Mm-hmm.
What is that? I think it's this
With
the
his
second
book.
one. Yeah.
Yeah, but like I wish that I just like I wish that I would have known like just how close you are to like having really cool research. Like everybody in academia, it's why I love that deep science ventures thing. I feel like a lot of PhD students and postdocs are like they're like right on the edge of like you're actually studying something very cool and you aren't looking at it from the right perspective. Right? Like delivery of siRNA could have been such I was like right there. I mean basically. Yeah, I don't know, man. Really it's really interesting to think about. So it's an exosome based delivery platform to deliver
Yeah.
P10 targeting siRNAs to basically promote nerve growth.
Yeah. So it's really interesting
Right.
to kind of compare contrast this with something like Matricel, right? Where you have basically the cells as the product inside of a matrix. It's basically just like sticking
Right.
the different blunt ends of the severed spinal cord back together.
Mhm.
And these guys are doing a bit more of a discrete approach where it's like, why isn't the body doing this itself already? Why
Right.
isn't it promoting this generation? Well, it turns out there's just this one gate that's locked, and if we can go in and smash the lock off that gate, we're going to allow this to go forward in a really positive direction.
Yeah. Kind of. Yes. It's for whatever reason like neuro regeneration is just like not a thing in humans.
Right.
Like it just doesn't exist. So you have like acute spinal cord injuries and you're basically screwed. Like what do you do? Or your neurons are going to grow back. But other kind of animals do. I feel like I remember seeing like people studying the regeneration of limbs in like, I don't know, those like lizard things. What are those called? Geckos? Whatever.
Sure.
Yeah. You know what I'm talking about. You can cut their tail off. Their tail grows back.
Oh, right. Yeah. I think
Right.
the geckos.
People study like the gene expression and like all the things and like the neuronal regeneration of those kind of species. That must not have that must not have gone very far because I haven't heard of anything. Although maybe like Matricelf was inspired by that. I don't know. Whatever. Um, but it is an interesting area of biology where it's like, hey, you lose neurons. Even like it would help us get smarter. Like I definitely lost a bunch of brain cells in college drinking. Like, can I have some kind of magic pill to make me smarter again? That'd be great.
Probably not. It's too far gone.
Yeah, this is way too far gone now. But anyways, Ben, I gotta jump to a noon meeting that got moved from 1. So if you'll
Excellent.
forgive me.
Not a problem.
Yes.
Very good. Well, ladies and gentlemen, thank you for joining us today. It has been a pleasure as always. It's awesome that we kind of get to do this as almost like for fun at this point. Although there are plans coming for the future inside of the Biotech Voyager. We're going to be doing special debates across different modalities. We'll be planning to bring you some live conference coverage in the coming months. So please stay tuned for that. And if you're not subscribed to the Biotech Voyager on Spotify, YouTube, or Apple Podcasts, please feel free to do so. We appreciate your time, and we'll see you next time. All right. Bye, everyone.